The next topic under the same chapter, human reproduction, is the cross section or the transverse section through a testis and ovaries. It's very important as it comes as spotters for practicals as well as direct questions in the theory. So, from both point of view, it's very important. So, let's go through the sectional view of the testis and the ovaries. Now, as we know, the testis, it's a pair of testes. So, there are a pair of testes. Each testis is divided into a number of lobules, 250 lobules, and these lobules are called seminiferous lobules. See, each testis is divided, radiating light, divided into a number of lobules. How many lobules? 250 lobules. These lobules are called seminiferous lobules. And before that, you remember that the testis is covered by a fibrous membrane called tunica albuginea. Tunica albuginea is a fibrous membrane that surrounds the testis. It's a pair inside the scrotum, and each testis is divided into 250 lobules called seminiferous lobules. And each seminiferous lobule, see, just look into the seminiferous lobule. Each seminiferous lobule consists of seminiferous tubules. See, which is highly coiled. There may be 3 seminiferous tubules in one lobule, and each seminiferous tubule, we are going to see the cross section of a seminiferous tubule. When we take a cross section, it appears to be circle. Each seminiferous tubule is lined by an epithelium called germinal epithelium, and this epithelium comprises 2 types of cells. The cells are spermatogonia. You are aware of spermatogonia, no? Interrupted by pyramid shaped cells called Sertoli cells. So, the seminiferous tubule is lined by a germinal epithelium which is indicated in white color. There are 2 types of cells, that is the the spermatogonia is the spherical one, spermatogonia, and the pyramid shaped cells are the Sertoli cells. Now, we know the function of the spermatogonia. It is a sperm mother cell which undergoes mitosis, then meiosis, and finally we get the sperms. We have learnt under the topic spermatogenesis. So, I am going to draw it like a stack, the different stages of spermatogenesis. So, this is, I am just zooming into one diagram, one portion. Consider this. I am taking this spermatogonia, this one spermatogonia. Now, what happens is spermatogonia, it undergoes mitosis to form the primary spermatocyte. Primary spermatocyte undergoes meiotic division 1 to form the secondary spermatocyte, and secondary spermatocyte undergoes a second meiotic division to form the spermatid, and then the sperms. So, you can see it in the different stages of development like a stack. Okay, so again spermatogonia, primary spermatocyte, secondary spermatocyte, spermatid, sperm. Again spermatogonia, primary spermatocyte, secondary spermatocyte, spermatid, sperm. But remember, the sperms is not located like this. All the sperms that are formed gets embedded in the Sertoli cells. So, these 3 sperms I am bringing it here. It gets embedded in the adjacent Sertoli cells. So, all the sperms that are formed as a result of spermatogenesis gets embedded. The head is embedded in the Sertoli cells. So, guess the function of the Sertoli cells. The Sertoli cells gives nourishment to the sperms. So, once again, the male reproductive organ is the testis. It is located outside the abdominal cavity in a sac called scrotum. Taking the cross section, there is a pair of testis, and it is covered by a fibrous membrane called tunica albuginea. Each testis is divided into 250 lobules called seminiferous lobules, and each lobule comprises of highly coiled seminiferous tubules. And each seminiferous tubule, when we take the cross section, is lined by an epithelium called germinal epithelium. This germinal epithelium comprises two types of cells. One type of cell is pyramid shaped. I'll highlight it with another color, pyramid shaped. And the other, between the adjacent pyramid shaped Sertoli cells, are the spermatogonia. And we have learned what is the role of spermatogonia. Spermatogonia gives rise to sperms we have learned by spermatogenesis. So we can see if this is the spermatogonia, this is the secondary primary spermatocyte, this is secondary spermatocyte, this is the spermatid, and we have the sperms. Primary, secondary spermatocytes, spermatid, and sperms. And remember, the sperms have their head embedded in the Sertoli cells. So the function of the Sertoli cells is to give nourishment to the developing sperms. Now coming out of the seminiferous tubule, this gap between adjacent seminiferous tubule, this gap is known as the interstitial space. See this gap, triangle almost, that intercellular space is called the interstitial space. There is a group of cells. See, I am drawing group of cells in this interstitial space. So the name of these cells are called the interstitial cells or the Leydig cells. These Leydig cells are very important. They are endocrine in function. They produce the hormone testosterone. Okay. So between the seminiferous tubules, between adjacent seminiferous tubules, there is a gap called the seminiferous, it's called the interstitial space. Inside this interstitial space are islands of cells or tissues. They are called the Leydig cells. The function of the Leydig cells is they produce the hormone testosterone, which is involved in spermatogenesis. It helps in the secondary growth of the secondary sexual characters, etc. Okay, so that is the structure of the testis. Now moving on. Now before that, through the slide under the microscope, this is how you are going to see the testis, a cross section to the cross section of the testis. So this is indicating the seminiferous tubules. So like you see number of seminiferous tubules and with interstitial space, and here in this space there is the Leydig cells, and we know what is inside the seminiferous tubules. So this is what we are going to see under the microscope, the light microscope. Next, moving on to the female reproductive system. Now the female primary sex organ is the ovaries. There is a pair of ovaries I have mentioned. It is on either sides of the uterus. See, on either side of the uterus, held to the uterine walls as well as to the pelvic wall with the help of ligaments. Okay, so there is the right and left ovaries. Now the ovaries, each ovary is surrounded by an epithelium. See, by an epithelium, and this epithelium is thin, it's called the germinal epithelium. Okay, now this ovary is divided into two zones. The two zones are usually most of our internal organs, like say adrenal glands or pituitary. Most of our glands or most of our organs are divided into two zones: outer cortex, inner medulla. So same way it is divided into outer cortex. If I am highlighting this as cortex, and inner medulla. And don't forget, it is covered by the germinal epithelium. So both the ovaries are surrounded by a thin germinal epithelium, and is divided into two zones: inner medulla and outer cortex. Now in the inner medulla, what we see is blood vessels, nerve fibers, and smooth muscle fibers. So they are present in the inner medulla, and in the outer cortex we see the ovarian follicles at various stages of development. Now looking into this, we can see the ovarian follicles at various stages of development. See, this is one ovarian follicle. If I am highlighting it here, these are the ovarian follicles at various stages of development. And look at the medulla. The medulla, I told you, it comprises what and all? Blood vessels, nerve fibers and smooth muscle fibers. Now we are going to learn the details of the ovarian follicles. Only after learning the different stages of the ovarian follicle, we can place it here. This is very helpful not only to study the structure of the ovary, also to study and understand the menstrual cycle. So I am going to give it in a horizontal representation, the different stages of the ovarian follicles. So first let me tell the names of the stages, very easy to remember the stages. Primary ovarian follicle, secondary ovarian follicle, tertiary ovarian follicle, Graafian follicle, and finally we have the ruptured Graafian follicle. Okay, so now I just gave a base here. Did you see I drew spheres? That's very important. It represents the oocyte. Now you have to connect it with oogenesis. We have learned in oogenesis the oogonia gives rise to the primary oocyte, primary oocyte gives rise to the secondary oocyte, and secondary oocyte gives rise to the egg. Now this now in white circle is indicating the oocyte, but remember which oocyte. Now in the primary ovarian follicle, this is the primary oocyte. In the secondary ovarian follicle also, this is the primary oocyte. But in the tertiary ovarian follicle, what must have happened to the primary oocyte? It underwent the first meiotic division to form the secondary oocyte. Now it is referred to as secondary oocyte. Similarly, in the Graafian follicle, what is the sphere indicating? Secondary oocyte. So when you analyze it, the oocyte of primary and secondary is primary oocyte, and the oocyte of tertiary and Graafian follicle is the secondary oocyte. So once again, I'll just highlight the different stages. Primary ovarian follicle, secondary ovarian follicle, tertiary ovarian follicle, Graafian follicle, ruptured Graafian follicle. So what did I teach you first? In primary ovarian follicle, what is this? Primary oocyte. Secondary ovarian follicle, what is the sphere? Again primary oocyte. Tertiary and Graafian follicle, what is this? Secondary oocyte. Then what will be the difference? I am going to draw further the structure. Now this oocyte is surrounded by follicular cells. So any oocyte, whether it is primary or secondary, it is surrounded by follicular cells. Follicular cells are granulosa cells. So what are these structures called? Follicle cells or granulosa. And in the center, what is present? As I told you, it's a primary oocyte. So this is the structure of a primary ovarian follicle. Clear? Next, secondary ovarian follicle. Again secondary ovarian follicle. What is the oocyte? The oocyte is again the primary oocyte. But now you look, what is the difference? Here you can see, look at the granulosa cells, one layer, two layer, and one more layer, three layers. So what's the difference between the first structure and second structure? The difference is this: in the first structure, on the primary ovarian follicle, there's only one layer of granulosa cells or follicular cells. Here there are many layers. But the similarity of both these structures is that the oocyte is the primary oocyte. So you got the clear picture of the distinction between primary oocyte and secondary oocyte. So from primary oocyte it proceeds to the secondary oocyte, that is extra granulosa layers are surrounding the primary oocyte. So that is done. The primary ovarian follicle is done. Next, tertiary ovarian follicle. Now, tertiary ovarian follicle, what is that structure? I am giving a lighter shade. That structure is not primary oocyte, it has become secondary oocyte. So, what must have happened? First meiotic division must have taken place. First meiotic division took place, and what is formed? Secondary oocyte and the polar body, first polar body. Anyway, I am erasing the polar body, but you should know the two cells that are formed as a result of the first meiotic division, secondary oocyte and the first polar body. So, this is secondary oocyte, here also this is secondary oocyte. Now, what happens here is the granulosa cells, look, gets organized in such a way that it incorporates a fluid filled cavity. A fluid filled, I'll indicate fluid with blue color, a fluid filled cavity called antrum. So, a fluid filled cavity is present and the granulosa cells get organized in such a way to accommodate this fluid filled cavity, and they get organized in such a way that it forms two thecas. Look, the theca that surrounds the antrum is known as theca interna, and the theca that is outside, the light pink, is called the theca externa. So, what is the difference of the tertiary ovarian follicle from the secondary ovarian follicle? One, a fluid filled cavity is formed. Two, the granulosa cells get organized in such a way to form two thecas, theca interna and theca externa. And three, the primary oocyte has undergone first meiotic division to form the secondary oocyte and the first polar body. So, there is lots of differences between the tertiary ovarian follicle and the secondary ovarian follicle. Next, what happens is the Graafian follicle. Now, for Graafian follicle, what I am going to do is, this diagram, I am going to replicate this diagram so that it will be instilled in your brain. That Graafian follicle is exactly equal to tertiary ovarian follicle. Then what is the difference? The difference is that in red I will indicate this secondary oocyte is surrounded by, I am indicating with green, secondary oocyte is surrounded by a layer called zona pellucida. So, the reason why I took an exact copy of tertiary ovarian follicle is that this should be instilled in your brain. What is Graafian follicle? It's exact description of the tertiary ovarian follicle. Only thing, an additional structure or membrane surrounds the secondary oocyte. What is that layer called? Zona pellucida. So, if you are asked to describe the Graafian follicle, give all the description related to the tertiary ovarian follicle. In addition to that, you must say that membrane surrounds the secondary oocyte called the zona pellucida. Now, last, what happens? The ruptured Graafian follicle. So, again, the last stage is the ruptured Graafian follicle. I am going to make use of this diagram itself. The Graafian follicles ruptures. See, Graafian follicle ruptures, and what happens? This egg is released, or sorry, the oocyte is released. The secondary oocyte is released. So, this is the representation of ruptured Graafian follicle. It's just simple as this. The granulosa cells ruptures and releases the secondary oocyte. This is release, secondary oocyte is release. So each month we say during menstruation, which I am going to take in the next topic, that one egg is released. Not egg actually, it is secondary oocyte that is released. The granulosa cells ruptures and release the egg or the secondary oocyte, which is usually said as egg, but in correct terms it is the secondary oocyte. This process is known as ovulation. Now we got a clear picture of the different stages of the ovarian follicle. Now these structures, I have to incorporate it into the peripheral part of the ovary, which is the cortex. So this is the primary ovarian follicle, this is secondary ovarian follicle, this is the tertiary ovarian follicle, this is the Graafian follicle, ruptured Graafian follicle, and the ruptured, I explained till ruptured Graafian follicle till the egg is released or the ovum is released, and that empty, see now this structure, it is an empty Graafian follicle, and this empty Graafian follicle is called corpus luteum. See, empty, it's yellowish in color, and very important, it has a function. You have to place it at the periphery, the different stages, that is in the cortex. Primary, secondary, tertiary, Graafian, ruptured Graafian follicle, empty Graafian follicle. What is empty Graafian follicle called? Corpus luteum. Okay, now I have to mention the endocrine part of the ovaries. Now we told there are two hormones, namely estrogen and progesterone. Now the source of estrogen are the follicular cells. So the estrogen are produced by the follicular cells. See here, estrogen is produced here, here more estrogen is produced, here maximum estrogen is produced. Now the source of progesterone is the corpus luteum. What is corpus luteum? The empty Graafian follicle, which is here. See, this is the source of progesterone. The yellow mass, which is not having the egg, that empty Graafian follicle is called corpus luteum, that only produces progesterone. Very, very important to maintain pregnancy. I hope you have understood the structure of the ovary.